Hi guys, it's Ellie. Today we're going to be discussing the cell cycle. So the first question we're asking about the cell cycle is, how is the cell cycle controlled by internal and external mechanisms? So to answer this question, first we have to understand what the cell cycle is in itself. And so the cell cycle is a complex set of stages that is highly regulated. And the whole goal of the cell cycle is for the cell to split into two identical daughter cells and go through cellular division. So the two main parts of the cell cycle are interphase and mitosis, and we're going to explain them in the next graphic here. So interphase is the first part of the cell cycle, and the cell spends 90% of its time in interphase. It is the largest part of the cell cycle. And so we divide it up into 4 subgroups, and we're just going to discuss these three and discuss this one later. But the first subunit of interphase is the G1, the GAP1 subunit, and in the GAP1 subunit, the cells increase in size, produce RNA, and synthesize protein. Then it moves on into the S subunit of interphase, which stands for synthesis. And in this portion of interphase, it is just a period of DNA replication. And then it moves on to the GAP2 portion of interphase, in which cells continue to grow in size, produce proteins, and prepare for mitosis, which is the M phase. In a mitosis, or the M phase, all energy of the cell is put and focused on the division of the singular cell into 2 identical daughter cells. Okay, so now that we've gone over the cell cycle into more depth describing interphase and mitosis, we're going to talk about these things in the cell cycle called checkpoints. And so checkpoints are the internal controls that direct the cell cycle. And each checkpoint checks for a certain crucial event to have occurred before the go signal is given for the cell to continue throughout the cellular cycle. And so the checkpoints that occur in the cell cycle, they act as built-in stop signals that halt the cell's movement throughout the cell cycle unless they're overridden by go-ahead signals. And so you're probably going to ask, how can the cell override the stop signals and get the go-ahead signal? And it's through its completion of these internal and external factors. And so we're going to discuss it more now. And so here we have another picture of the cell cycle. It's a little more in-depth because it includes the checkpoints. And so, like we stated before, the checkpoints check for certain crucial events to have occurred before the cell can continue throughout the cycle. And so once it hits this G1 checkpoint, the GAP1 checkpoint, the factors it's looking for the cell to have completed, or the factors that this is looking for the cell to have gone through, it's looking to make sure the cell is big enough to continue and that the environment is stable enough for the cell to continue the cell cycle. So if the cell passes this test and answers these questions correctly, it will move on into the G2 checkpoint. It will go through synthesis and go into the GAP2 checkpoint. In the GAP2 checkpoint is making sure that the DNA in the cells is replicated correctly. Again, that the cell is big enough and that the environment of the cell is stable enough for it to continue to mitosis. Where it hits the third checkpoint, the M checkpoint. And the M checkpoint is checking to make sure that the chromosomes are aligned correctly on the spindle for the division of the cells to occur. And so you may be wondering, what happens if the cell does not get the go-ahead signal? What if the cell isn't big enough? What if the environment isn't stable enough? And what if it doesn't have the correct internal factors for the cell to continue through the cell cycle? And that's where this G0 part of interphase comes in. And the G0 phase is what happens if the cell does not get the appropriate signal, if it does not pass the checkpoint. And the cell will just kind of stay here. It will hang out here. It exits the cycle, hangs out here, and becomes a non-dividing cell, so it can't go through mitosis and divide. It can either stay here for a short amount of time until it kind of gets itself together, or it can stay here permanently. It just depends on the cell. All right, so now we're going to revisit this question. How is the cell cycle controlled by internal and external mechanisms? And we now know the answer to this question because we know that these internal and external factors are what allows the cell to pass the checkpoint. To finish up with this question, there's one more topic to cover. Right here we're looking at a cyclin, and a cyclin is a protein. The reason it's called a cyclin is because their levels cyclically in a cycle rise and fall in dividing cells, so it kind of creates this cycle in the cells. And then we have a kinase, and a kinase is a class of proteins that activates other proteins through phosphorylation. And these kinases are critical to the cell cycle, but they're only activated when they bind to a cyclin. So that's what we have here, and that's what's called a cyclin-dependent kinase. So it's dependent on this cyclin for it to work. And once they bond together, they form a cyclin CDK complex. And so now we're going to discuss a specific cyclin CDK complex. This one's called MPF, which is short for maturation promoting factor. But an easier way to think of it is the M phase promoting factor, because MPF triggers the cell's passage from the G2 phase into the M phase. And so without it, the cell cannot pass this G2 checkpoint to go into the M phase. And this graphic is kind of just describing how the cyclin CDK complexes are formed and then break apart. So basically the CDK is in the cell cycle, the cyclin is accumulated here, and then they bond together to create the complex, and now it's MPF, and it allows the cell to pass the checkpoint and go into mitosis. And then after the cell gets through mitosis, the cyclin degrades, the protein degrades, and leaves the cell cycle, while the CDK, the kinase, goes right back into the cell cycle and can be recycled. And so these cyclin CDK complexes are another internal factor that controls the cell cycle and makes it go around correctly. So now that we've discussed all the internal and external factors that control the cell cycle and make it go around correctly, we're going to talk about what happens if the cell cycle doesn't go correctly. One question we're going to answer in this video is, how can the loss of control in the cell cycle lead to cancer? And so to answer this question, we have to understand what a growth factor is. And a growth factor basically just controls the timing of the cell. So there are positive growth factors that allow the cell to continue to divide, and then there are negative growth factors that stop the cell from dividing. And so we're going to talk about a positive growth factor right now. It's called a PDGF, which stands for platelet-derived growth factor. And the PDGF, it is a growth factor that is made by blood cells, like you can see right here. This is the blood cell. And it is required for the division of fibroblasts. And fibroblasts is an important connective tissue in our body. And so without this PDGF, the fibroblasts would not be able to be divided and made into cells. And without fibroblasts, we cannot be able to heal from like cuts and stuff like that. So without this PDGF, without this growth factor, we would not be able to heal properly. And so back to this graphic again. So now that we've learned about PDGFs, that positive growth factor that allows the fibroblasts to be created, we know that helps the cells divide. But the issue with cancer cells is, whether it's a positive growth factor or a negative growth factor, the cell cannot differentiate. We'll just continue to divide. And basically how a cancer cell is created is basically that most cancers arise as cells acquire mutations that make them divide more quickly and escape those internal and external factors that control the cell. And so this can happen, as seen in this graphic, as the cell continues to go through the cell cycle and be divided into two different cells. It starts with one mutation, such as this initial mutation, divides into two cells. This cell gets the mutation again, divides into two cells. Another mutation, then all these mutations allow the cell to completely evade these internal and external controls that allow it to go through the checkpoints. So now the cell is passing these checkpoints without, going through all the factors that it's required to do. So like, for example, when it goes through gap 1, which is asking, is the cell big enough, is the environment stable? The cell may not be big enough, or the environment may not be stable enough, but it will be able to pass this checkpoint due to its mutation, which makes the cell duplicate, or not duplicate, it will make it divide irregularly, which will lead to this rapid division of cells, which is the cause of cancer. So just a quick recap. Now we know that the cell cycle is what leads to the division of the cell into the two identical daughter cells. We know that it has the two parts, interphase and mitosis. We know that it goes through checkpoints to be able to be regulated, and these internal and external factors are regulating these checkpoints to be making sure that the cell goes through the cell cycle properly. And now we know that certain mutations that occur in cells can lead to the cell not going through the cell cycle properly, which leads to the formation of cancer. So thank you guys so much, and yeah.